Okay, I think we should all be here. So, yeah, hello everyone. Good morning, good afternoon, good night, depending on where you are. Thanks for joining us on today's installment of Paleo Perks. Today we'll be hearing from Lazaro Vinola, who's based at the Field Museum of Natural History in the United States, and he'll be talking about Sebecids in the Caribbean and other unexpected discoveries from the islands. So a quick rundown of the format of today's seminar. We'll start out right now with some brief welcome and announcements. The talk should last about 30 minutes. After the talk, we'll have a moderated question and answer session. And during the question and answer session, you can send your questions directly to the Questions at Paleo Perks host, who today is going to be Neha Sharma. And then afterwards, our speaker has agreed to join us for tea time. So that's just a more relaxed, casual setting to talk about scientific topics and non-scientific topics. So if you enjoy the talk and want to chat a little more, feel free to stick around. A few things about Paleo Perks housekeeping. Paleo Perks values the participation of all folks interested in the paleo sciences. Please remember to abide by our code of conduct during today's seminar. Please mute yourself during the duration of your talk. If you find yourself unmuted, please make the effort to mute yourself out of respect for the speaker and for the other people in the audience. You can ask questions by chatting to the Questions at Paleo Perks host, who again today is going to be Neha. Or alternatively, you can use the raise hand function. Additionally, technical issues should be sent to the Questions at Paleo Perks host. We have closed captions built into Zoom. You can use the CC button to show or to hide them. Additionally, we are always on the lookout for outstanding early career researchers who could potentially give a Paleo Perks talk. So you can nominate an early career researcher with this link here, which will be shared in the chat shortly. Additionally, we collect weekly feedback for demographic information. It's completely anonymous and it's completely optional, but it is encouraged as it helps us shape these seminars to best serve the paleo community. And you can also find that link briefly in the chat window. Now on to our speaker. Lazaro Vinola did his bachelor's in paleontology at Montana State University in the US, and then went on to do a PhD in paleontology at the University of Florida, also in the USA. Currently, he is a postdoctoral fellow at the Field Museum of Natural History, also in the United States. So with that, I will hand over the reins to Lazaro, and yeah, take it away.
Thank you so much. And let me start with sharing the presentation for today. Okay. Can you see well?
Yep, looks good.
Okay. And thank you everyone for coming today and for the invitation to participate in Paleo Perks. I'm very excited. So I'm going to be talking today about Sebecids in the Caribbean, which is like these ancient crocs from South America, and other unexpected discoveries that we have done in the last few years while working on the islands, including some strange pigs that tell the story of the origin and evolution of parrotfishes, and also some bone-loving insects that we discovered in some cave in Dominican Republic. I am originally from Cuba, where I grew up in the south, around 2 hours away from Havana. And as a kid, I was a very odd one, always looking for bones, going into cave and quarries and collecting fossils as a little kid. So I used to have like a large collection when I would go like every summer to the Museum of Natural History. So since a little kid, I was in love with it. And just to give you a context, most of my work so far has been in the Caribbean. And the Caribbean is divided to the east of this lies on the Lesser Antilles. To the north of the yellow line is the Bahamas, and to the south are the Great Antilles. So the Great Antilles, the large islands, are Cuba, Hispaniola, and Puerto Rico, and that is where most of my works have focused so far. In the Caribbean, we have the oldest fossils dating from the early late Jurassic, from Orphean deposits in western Cuba, including ichthyosaurs, pterosaurs, and also some dinosaurs remains. The first islands that we have in the Caribbean with fossils for vertebrates are from the late Cretaceous. And at this point, it was a group of a chain of volcanic islands in what is today Central America. Then we have the formation of the first permanent islands in the Caribbean in the Eocene. But most of the fossil record that we have from the Caribbean is actually from the very late Cenozoic. If we look specifically in the Cenozoic, you will see that it's again dominated by quaternary deposits. For example, in the island of Cuba, we have around 215 deposit known. They're mostly cave and sinkhole deposits. Hispaniolas have at least 124, but there are more. Well, in the Miocene, Oligocene, and Eocene, the number of locality with fossils is much lower, and the quantity of fossils that we have recorded from there is also much smaller. The fauna today in the Caribbean, the predator guild, is dominated by raptorial birds, snakes, and crocodiles. And if we look at the fossil record, the quaternary one, we start seeing the same. We have the Cuban crocodile that is today restricted to Cuba. It's also found in the Bahamas, Cayman Island, and Hispaniola. Then we have giant owls like Onymegalonyx oteroi, nearly reaching 1 meter of height from Cuba. And then we also have very large hawks that were endemic from the Caribbean. There is entire radiation there. So the fauna from the Caribbean islands that included sloth and tortoises, and the rest of range of like flylet birds was dominated again by these secondary predators that evolved to have these large body sizes. And this is very characteristic of oceanic islands. That means that they have been islands that have been isolated since the origin. And for this reason, there have been some researchers that have proposed that the Caribbean islands were never connected with other land masses, and the fauna have reached there by oil water dispersal. Now, the question is that we don't know too much about how was the fauna before the late Quaternary. And these same patterns that we're seeing them were present millions of years ago. So the first insight about the fauna before the late Quaternary came from the deposit that was called Domo de Sasa. That is around 18.5 to 17.5 million years ago, early Miocene. And it was discovered in central Cuba in the 90s when they were building a dam. So this deposit, they cut a little hill and they exposed several fossils, including some of the oldest sloth at the moment, the oldest primate and rodent discovered in the Caribbean at that point. But besides this fauna, they also discovered a suite of fossil crocodilians, which later in 2014 were published by Christopher Broshu, expert in crocodilians, and the Cuban paleontologist Osvaldo Jimenez. The paper focuses on multiple fossils that they have discovered in that locality, including these compressed teeth, these cephalodon teeth that were serrated. Now, these teeth looks like dinosaurs, theropod teeth, and it's because they belong to a carnivorous crocodilian. And this kind of morphology is present in three groups of crocodiles, crocodilians. Ones are the Sebecids. It's a family endemic from South America, and they were present there in the Cenozoic. The second one are planocranians, which are these like terrestrial crocodiles with hoof from North America, Europe, and Asia. And they were present in the Paleocene. And then you have the Megazuchians that are from Australia, and specifically Quinquana, which is from the late Quaternary, from the Quaternary, sorry. So this kind of teeth evolved at these three times in crocodilians, and it was difficult at the moment to assign the teeth along to one of the three groups. They discussed in the paper that it could likely belong to a Sebecid, but they haven't been found so far in the Caribbean. A few years later, in 2018, we did an expedition to Puerto Rico to some early Oligocene deposit, and we found another of these tooth. Again, compressed with serration, relatively small, but we have more material. It was nearly impossible to say to what it belonged to. So in 2019, also started working in some late Miocene, early Pliocene deposits in Hispaniola. Hispaniola is the second largest island in the Caribbean, and at this point, it was divided in multiple Paleo islands in the Miocene. Specifically, we're working in a locality in the northeast of the island that is known as Paleo Pond 1 and Paleo Pond 2. These two localities were discovered while building a highway, which led to finding some fossil sloth, eggshells, turtles, and a series of other impressive fossils. And of course, we found tooth, a serrated tooth, a Siphodon tooth. So by this moment, we have at least 4 different teeth from the 3 main islands in the Caribbean, ranging from 29 million years ago in the early Oligocene of Puerto Rico to 5 million years ago in the late Miocene and Pliocene of Hispaniola. Now, the key finding from this deposit were the vertebrates. And these are 2 vertebrates, a cervical one on the top and a caudal one on the bottom. And these vertebrates were the key because for Planocrani and Quinquana, they're in one clade of crocodilians are called Eosuchians. While Sebecids are like the primitive ancestors, the cousins, they're in another clade called Notosuchians. Eosuchians having the vertebra in the centrum, this ball and socket structure, while in the Notosuchians, they're flat. So in this way, we're able to assign the vertebrae from Dominican Republic to Sebecids specifically, and finally tie, the teeth with the vertebrae and figure out what kind of animal was present there. The interesting thing is that Sebecids so far were only known from South America, and they were among the apex predators during the Cenozoic, while South America was isolated from the rest of the continents. So they were sharing the top of the food chain with like Porosity, like the giant terror bird, with some of the carnivores, marsupials, and some of the giant snakes from South America. In the Paleogene, Sebecids were restricted were widespread throughout the continent, while in the Neogene, they were restricted to the northern part only. And this is probably associated with major climatic changes in South America and the reduction of the tropical region there. The question then remains how Sebecids arrived to the Caribbean. And there is a hypothesis that was put forward in the 90s that likely part of Cuba was connected, part of Cuba, Hispaniola, and Puerto Rico was connected with South America through how the Aves Reach, with this crest that is today underwater. And there have been some more recent work that is stating that probably some of like the Lesser Antilles also play a role and were exposed at this time around 33 to 35 million years ago. This is also consistent with the findings of some probably Sebecids also from Puerto Rico, the tooth that I mentioned before, which is around 29 million years ago. And it will make sense that the first Sebecids to be in the Caribbean will be in Puerto Rico, since it's the region more closer in the Great Antilles to that connection. So this locality is not only important because of discovering the first Sebecids, which again shows that there was an apex predator in the Caribbean, but we're also finding a very distinct fauna from there, including some of the fossils that we're currently working and will be published soon. And these include like some gharials also from there, from the late Miocene, early Oligocene. So stay tuned, and you will see probably something coming about this locality soon. And I will be heading there in early October to do more field work and do more fossil collecting. So besides these ones, another thing that I really liked when I was a kid was going to cave to collect fossils. And while going in caves, sometimes you not only find the late Quaternary fossils on the floor, but if you look to the roof, then you start finding all the things that were dissolving in the limestone, including Miocene fossils from fishes. So I used to go there and find sharks and rays and triggerfishes. But something that we collected and for a long time we didn't pay attention were actually parrotfishes. And parrotfishes are this fascinating and very diverse group of fishes. They have a pan-tropical distribution. They're very colorful. They're mostly associated with coral reef and seagrasses bed, and they are like among the more colorful things that when you go scuba diving you will be seeing in a coral reef. Parrotfishes have some special adaptations to these lifestyles in coral reef. They have a very derived beak that comes from the teeth conversion to form this beak-like structure, and then they have also very derived version plate that are used for grinding the corals and some of the other things that they consume. So these teeth and these version plates are very, very diagnostic for when you find them in the fossil record. And please, if you find them, if you know someone that have found them, let me know or like work with them because they're extremely interesting and extremely rare also in the fossil record. Now, parrotfishes are also very important because there are ecosystem engineers today. So they manage microalgae control, they create substrate for the growth of coral, they produce bioerosion, they produce sediment, and they're key for coral reef resilience. It has been found that when there is overfishing of these parrotfishes in some of the coral reef, they start losing some of the quality and quantity of their coral reef. And also, they're extremely important, and I put this photo there because for some species, a single parrotfish can produce up to 100 kilograms of sand. So if you like sandy beaches, coral reef parrotfishes are extremely important. They produce a lot of it. Now, most of the biogeography and origin of the parrotfishes have been based on molecular phylogenies. And most of the diversity of them is from the Pacific, from the Indo-Pacific. And the biogeographic analysis shows that only in the Atlantic you have the Spadisoma and close relative clay that arrived around 23 million years ago, probably from the Central Tethys. And then you have a later colonization by Scarus, which is one of the most diverse genus, around 6.5 million years ago. This diversity is significantly lower compared with the Pacific one that is considered to be the center of radiation and evolution, and where most of the clays dispersed from. Again, parrotfishes evolved around 30 million years ago in the late Oligocene, early Miocene, while most of the diversification occurred throughout the Miocene and Pliocene. The fossil record, as I mentioned before, is very sparse. There are only a handful of fossils, most of them very fragmentary. The most complete one is a Callostomus, and this is the specimen or part of the specimen that is from the middle to late Miocene on Europe. Now, we start looking at some of the fossils that we have gathered from Cuba and from some of the other places, and we start finding that several of the taxa that are alive today had never been reported in the fossil record before. And these include Scarus on the top, Spadisoma in the middle, that there was a record before in the Eastern Atlantic, but this was the first record for the Caribbean. And also another genus that is Nicolsina, only present in the Caribbean and the Eastern Pacific. There was no fossil record so far. So by itself, it was an interesting contribution to have all these taxa recorded for the first time and be able to help later on, like calibrate some of the phylogeny. Now, the very interesting discovery came from some of those fossils that I had collected as a kid in Cuba in those caves. And the plates are on the left. And these are like some of the plates part of the beak that I showed earlier on. And when I start comparing them, and also some fossils that were discovered in Florida, including the ones on the right, they actually belong to this. And this is a genus called Bulbometopon. It doesn't belong to the genus itself. It is close relative and extinct one. But the most interesting part is that this group is endemic from the Pacific. It's one of those clades that only evolved in the Pacific so far, or people thought so, and they never dispersed outside there. And at the same time, this corresponds to the oldest fossil of a parrotfish known so far. So with this discovery, we're able to change a little bit the biogeographical history of parrotfishes, showing that most likely that story about the Pacific as an epicenter of diversification wasn't the case. It's most likely that parrotfishes originated and diversified in the Mediterranean. From then they dispersed to the Pacific, to the Caribbean, and then with subsequent extinctions in the Caribbean is where you have that biodiversity patterns that you resulted today. More importantly, these teeth already had a derived morphology on this kind of beak that also shows that parrotfishes around 20 million years ago were already important elements in coral reef. So Bobo and Metopon and Cetuscatus, which are the ones close related with the fossils that we found in Cuba, have a specialized ecology where they are the biggest ones and they come and they break part of the coral reef and they grind it. Most of the other species are like smaller pieces. And this kind of ecology is represented in the Caribbean only by one species. And it's probably a convergent evolution after the extinction of these giant parrotfishes in the Caribbean. And again, this shows how sometimes some of these groups that we don't pay too much attention to, or that we think that they're fragmentary small fossils, actually tell a very interesting and important story for biogeography, but also for ecology. Lastly, I want to share with you another discovery that we did in Hispaniola, this case, and it is about bone-loving insects. And the Lake Quaternary, there is hundreds of localities in some of the islands, and there are thousands, if not hundreds of thousands of fossils. You go to some of the museums and you find drawers and drawers and drawers of specimens that haven't been cataloged, identified, and including sometimes new taxa of vertebrates. But the issue is that we don't have a very good idea about what was the insect diversity in the Lake Quaternary because it doesn't fossilize well. We have the amber fossils from the early Miocene of Dominican Republic that are very diverse, but from the Lake Quaternary, it's almost unknown. And as I said, the Caribbean islands, they're mostly limestone. Some of the islands like Cuba can be over 70% of limestone. Hispaniola is a similar case. And if you look at the south of the island of Hispaniola in Pedernales, for example, which is one of the areas that we work, and this is a satellite image, and then you pay attention, you see all those, tiny dots. Those are giant sinkholes. Some of those sinkholes can have like more than 100-meter wide. And if you're in the ground, it's not only these giant sinkholes, but there are cave and smaller sinkholes everywhere. You walk 20 meters in any direction, you will likely find one. And when you get down, some of them won't have fossils, but a lot of them will have it. And one of these cases was Cueva de Mono. It was a cave discovered more than 15 years ago, relatively small, have probably 15-meter long. There is at the end of the cave, at the distal part, a deposit with multiple layers of sedimentary fossils separated by carbonate layers, representing periods of high pluviosity of rain. And most of the deposit was originated by an owl. We have found fossils of the owls, of the juveniles, even of the eggshells, and they belong to an extinct giant owl named Tyro ostologa that was endemic from Hispaniola. There's also some fossils that belong to sloth and crocodiles and likely belong to animals that fell inside the cave or were remains that were around and during the storm were carried out inside the cave. Again, when I said that we find a lot of fossils, it's like this. This cave is the most productive one in Dominican Republic so far. We got more than 60 bags of sediment from this deposit, and there is still probably the same amount of material or double in the locality. It represents a very diverse fauna with over more than 50 species of vertebrates, including some new giant eagles from Hispaniola, some caracaras, undescribed rodents. And for my dissertation, I was focusing primarily on the diversity of rodents from the Caribbean. So this was like paradise for me. And there were some species, like in this case, which is like Plagiodon diarrheum, that we have beautiful skulls. And that species, the holotype, was only an upper premolar. And until they discovered this cave, there were only a handful of mandibles. And now we have complete ontogenetic series of these species. So it was an incredible discovery. But when we were in the locality and we were looking at some of the skulls and mandibles, and this is like a palette of one of the Plagiodontiarium, we start seeing these like concave structures on the sediment. And these are rodents with ever-growing teeth. So with the teeth, the animal decomposed after, I brought it to the deposit, the teeth fell out, and they were filled by sediment often. Not all the case, the skull here have like some teeth in place, the palate didn't have it. So in the sediment that was filling the alveoli, there were this chamber there. And we were finding not only that on the palates, we also start finding it on the mandibles. We start finding in some of the vertebrae of rodents. We found it in a pulse socket pulp of the molar of an sloth, and in many bony structures that resembles like a chamber. We start finding these little things, and that reminds me a lot of something that I was seeing in Montana when I first visited and I was collecting fossils there, and those were wasp nests. And it was thanks to Tony Martin, the paleontologist, who was like showing us these specimens and seeing how to identify them. Interesting thing is that we start looking, and usually wasp nests don't preserve. You usually preserve the cocoons. So the cousins of wasp are bees, and bees preserve the nest. The preservation of the nest actually comes from the fact that bees create an inner layer of the wall of the chamber, and this is done to put the food inside, which is usually some plant derived. So they wanted to make the chamber watertight, and these chambers are actually nests for the bees. And in this case, they belong to solitary bees, most likely, that were nesting on the ground. And most of the bees today do that. We usually think of them as like social, having these big colonies, segregate and work. But actually, most of the diversity of bees are not eusocial. They're solitary, or at least some degree of solidarity. And many of them build nests on the ground. Again, solitary bees, very pretty, awesome. When we start looking at the fossils from the DR, and we did some CT scanning from the specimens to try to estimate the size of the chamber to have a better idea of what it could belong to. We start finding also that in the alveoli, there was not only a single nest. We have, for example, this mandible of Plagiodontiarium, that each of the alveolus for the premolars and molar were filled with sediment, and they had chambers inside some of these nests. And a single alveolus, in some of the cases, have up to 6 generations of nests, showing that they were most likely coming to the same place and nesting one against and once against and once against, and not only using the same deposit, but also using the same chambers that they were using before. So most likely it's a solitary bee that is doing swarm nesting, so you have large groups of them coming and nesting, and they have some fidelity and coming back to the site. We were able to also double tie that was double check that were bees using electronic scanning microscopes and looking at the inner walls of these nests, and showing that actually they have a double packing wall, which will actually increase effectivity to make it watertight and the food will preserve inside. We'll try to look for pollen grains to try to identify some of the plants that these bees were using and were breeding for the larvae, but we couldn't find them. There was, I think, only like one pollen grain, and most likely it's because like the larvae actually developed it and ate it and left, which suggests that it was like probably a very good place for nesting, which makes sense as they were using for generation. But also we start finding a lot of evidence for microbial activity and fungal activity, probably after the bee left the nest. Probably this fungal and microbial activity will have destroyed some of the pollen that was there, and also will have restricted a little bit the walls of the nest, which we find evidence of like these tubes connecting. So we were able to, with some artists from Cuba, do some reconstruction of how the cave would have looked like several thousand years ago, and document the first instances of bee nesting in cave, and not only nesting in cave, but also using bones as chambers to contain the nest. Nothing like that has been documented before. They usually nest on the surface, on the ground. It seems that in this particular place in southern Hispaniola, they're using cave partially because most of the exterior is limestone and there is no very deep soil deposits there. So instead of having where to nest, they usually go to the cave where most of the soil is being deposited. How they become to using bone chambers as nesting structure is unknown at the moment, but it at least expands what we know as a behavior for ground nesting bees. And I think I went a little bit fast, but with this, I want to thank everyone that have helped me throughout these projects. And this is not work that I have done alone. This is work that I have done with like a very large team of friends and collaborators, including people from Cuba, Hispaniola, France, Puerto Rico, Australia, from all over the world. And most of this work was collateral to my dissertation, which was working on rodents. And these were sort of like the fun, interesting projects that we were doing while working in the Caribbean. And the things that we're sometimes, finding on the side that actually sometimes were as interesting as the other things that I was focusing in. I also want to thank all the institutions, including museums and organizations that gave me funding and support to do field work and to do all this research. So also thank you so much for Paleoperks for inviting me to give this presentation and for everyone to come in and listening to the presentation. Thank you.
Great. Thank you so much, Lazaro, for that really interesting talk. Just a reminder to, a reminder to the people in the audience that if you have questions, you can send those to the questions at Paleoperks host for our moderated question and answer session. But yeah, very interesting talk, Lazaro. We already have a couple of questions that have come in.
Thanks.
And so we can just hop right into the Q&A if you're ready for it.
Let's go for it. Yeah.
All right. Sounds good. So our first question, which I'm going to put in the chat. Do such biological interactions between insects and vertebrates still exist today? So I suppose that means in kind of the modern. If so, do you foresee those interactions changing with climate change, potentially?
So we're trying to figure out what was the bee that was doing this nest. We weren't able to do it. There is no bee in Hispaniola that have this kind of behavior. There's only one documented case of a bee from Cuba that was found nesting in the entrance of a cave. But outside of that, it hasn't documented. And again, the issue also is that the nesting ecology of many bee species, especially endemic to the islands, is poorly known. So it could be that it's a species that is still alive on Hispaniola. It could be that it's still alive around the cave. We put traps to try to get some of the diversity of bees in the cave, and it didn't belong to any of those. And also, there is a possibility that is was a very specialized bee that was used in, these vertebrate remains for nesting. Now, bear in mind that these vertebrate remains belong to extinct species. So this nesting structure doesn't exist anymore today. So if it was very specialized on that kind of structure, it could also be the case that it belonged to an extinct species of bee. And this is the only way that we're documenting them. Yeah.
Yeah, great. Thank you so much for that answer.
Yeah.
And thank you for the question. Okay, we have another one. Okay. And this question reads, what is your dream field site that you would love to study if you could? So I guess assuming, unlimited funding and resources.
Ooh, thanks. That is a really good question. Um, I have to stick to the Caribbean. I love it there, and I think there's so many interesting things that will come from there. So the place in Hispaniola that we're working now is very close to a dream place because we're finding so much interesting fauna from there that we didn't even have an idea it was going to be that case. Another dream place would be some of the late Cretaceous deposits in central Cuba that they document some of the first islands that were present in the Caribbean. And we found some pterosaurs, some plants from there. And this is one of my dream places, because at that point, the Caribbean was in what is today Central America. It was a large series of islands. And in the late Cretaceous, you have the exchange of dinosaurs and mammals and crocodilians and other groups between South America and North America, which were isolated at that point. So there always been the question where, like, how the animals, cross it. And one of the hypotheses that it did throughout Cuba. And we found fossils of the plant that belong to one of the groups that cross it. So I think that potentially they could be some of the ancestors of marsupials from South America could have gone throughout Cuba, and some of the early dinosaurs that also crossed and gave origin to some of the hadrosaurs from South America could have crossed throughout Cuba. So I think that would be a perfect place to go and find them. Yeah. But thank you so much for the question. Yeah.
Yeah, good answer. That's very interesting. A lot of potential, I think.
Yes.
And that actually blends nicely with the next question, which is, maybe a bit open-ended, but what do you see as the next steps for this work, more specifically?
Okay. Thank that is a very good question too. Uh, so for the work that we're doing right now in Dominican Republic in the late Miocene, early Pliocene, the idea is to keep expanding in the collecting that we're doing. So we're doing some of the classic quarrying, also some of the screen washing, and we're trying to document most of the diversity that is present in this locality. And this is turning out to be pretty important because it's showing that only 5 million years ago, the diversity that we're finding in the Caribbean is very different from what we find in Lake Quaternary or today. So what we understand as this like hotspot of biodiversity and the classic Caribbean diversity, you know, maybe something relatively recent, like talking in geologic time. So we're trying to focus on these localities. We're trying to discover some more deposits a little bit older and a little bit younger to try to understand what is the timing of extinction and fauna overturn in the Caribbean in this pre-quaternary deposit. At the same time, there is a lot of work that has to be done with the late quaternary deposits across the island. Partially, most of the mammals have been well studied or sort of. There's still a lot of work to do, but there are other taxonomic groups like birds, amphibians, and reptiles that have been very little work. So there's an open field to keep working there, to potentially have like students coming and also collaborating and keep expanding on what has been done. Thanks.
Great, awesome answer. Thank you for elaborating on that. So just a reminder to our audience members that if you have any questions, you should please submit those now because we're actually down to our last submitted question, which I will share. But yeah, if you're in the audience and you have a question, please send it our way. Okay, I'm going to place this in the chat. This question reads, what are the biggest challenges with this work? Does weathering and wetter climate impact the specimens
A
or not?
lot.
Hmm.
Yes, thanks. A lot. Like working in the tropic can be very challenging. Like I would love that would be like the backlands when you go and everything is exposed and you come and you collect. Like some of these localities can last like a very short time. Sometimes they're fortuitous, like the deposit in Hispaniola, they were building a highway and they cut through a hill, and that is how the locality got discovered. But in a couple of months to years, they can get eroded and they can get covered by vegetation, making the work in the Caribbean very difficult. Some of the fossils that we have gone, derive and they're like partially eroding away. So that can be problematic. And sometimes, like always, getting like funding and support to go over to another country to do collecting, to do processing, that can be challenging sometimes. But you know, I think it's worth trying to do this kind of work. It's important also to do it, and that also make it time sensitive. Since we have some of these localities that are like going away, there's an emergency and there is a necessity to go and try to rescue as many fossils as possible while we can. Thanks.
Yeah, thank you. Very, very interesting. Um, so with that, unless anyone else in the audience has some final questions, we can wrap up the question and answer session and take a quick break before tea time. So I will share my screen. Okay, yeah, so thank you again to our speaker for the really interesting talk and for the question and answer session. And thank you to everyone who was in attendance today. Again, please fill out the weekly feedback form so that we can learn a bit more about who attended today's seminar, as well as the speaker nomination form, which I will share in the chat briefly. And just a reminder about our seminar next week on the 30th of September. We'll be hearing from Charlotte Hipkiss from the British Geological Survey in the UK about paleoclimate reconstruction. So we look forward to seeing you all then. And now we will have a tea time, which is just an informal conversation about the talk and about any other themes that people would like to talk about in a more relaxed setting. So now is a time where we will have a little break so you can get up, walk around, have a drink of water, and we'll meet back here in 2 minutes for those who are interested in sticking around for tea time. If you have paleo pets, feel free to bring them. And yeah, thank you all for attending, and I will see some of you back here in 2 minutes. All right, bye-bye.